Now, hopefully you've watched the video on transcription, and this is the next stage that follows along. This is the stage of RNA processing. So when we finished transcription, we were left with this molecule called pre-mRNA. Now, pre-mRNA is a single-stranded molecule of RNA. So we've got uracil instead of thymine, and we have ribose sugar in the backbone instead of deoxyribose sugar in the backbone. So when we look at this, we're now looking at a stage that kind of happens round about here. Yeah, so we're still in the nucleus. Sometimes I believe it can happen outside, but most of the time we kind of float around in the nucleus, and we are going to process that mRNA, that pre-mRNA, to make it into what we call mature mRNA. So this is a really simplistic picture that shows you what it looks like kind of before and what it looks like afterwards. But I'm going to add in a little diagram at the top. So when you finished transcription, what you end up with is a molecule that looks a bit like this. Yeah, it's just a whole bunch of RNA nucleotides, and this is what we call pre-mRNA. Right, so that pre-mRNA has a tendency to break down. It has a tendency to degrade. There's also a whole bunch of information in it that we don't need to turn into a protein. So we're going to do three main things. So there were 4 things in transcription, and there are 3 main parts of RNA processing and why they occur. All right, so the first thing we're going to do is we're going to add what we call a methyl or a 5' cap to one end of the strand. If you want to know, it's the 5' end, but you aren't required to understand 5' and 3', so you can just leave that out if you wish. On the other end of our piece of mRNA, we're going to add a whole bunch of adenine nucleotides. So this is what we call a poly-A tail. So when you're talking about what happens during RNA processing, we're adding a methyl cap to one end and a poly-A tail to the other end. After you've done that, or concurrently while that's happening, there's also this process of splicing. Now, this one here has been anywhere between a 1 to 4 mark exam question just on the process of splicing. And I know 4 marks seems a bit scary, but when we break it down, you can see why. So the first thing that happens is there are sections of DNA called introns within the mRNA. Now, these are what we call non-coding DNA. I want us to stay away from the word junk, but just call it non-coding DNA. And the big thing here is you would say introns are removed because they are non-coding pieces of DNA. So that's 2 marks just there, yeah? Introns are removed because they are non-coding pieces of DNA, or non-coding RNA. That I'm going to change that there. So I'm going to say they're non-coding RNA, and they're removed from this piece of pre-mRNA. Now, what you get left with are these things called exons. And exons are the bits of RNA that actually code for a protein. So they are joined together. So exons are kept, yes, because they are expressed. So exons are expressed. So that's why they are kept. Exons expressed, and introns are intruding pieces of DNA that we want to remove. So introns are intruding and they're removed. I know it seems a little bit counterintuitive. You feel like the introns should be the ones kept and the exons should be the ones removed. However, that's not the case. So the exons are the bits of the RNA that we keep. The introns are the bits of the RNA that we throw away. And then after you've done that, you've added your poly-A tail and you've added your methyl cap, and that's about it for splicing. So you can see another picture here just to show us what's happening. So here's another way of remembering it. We've got introns are the non-coding or intervening or intruding sequences, and exons are the expressing sequences within the protein. So you can see here we have a string of DNA at number 1, and it has exons and introns, exons and introns, exons and introns. What happens to that is we have this thing called a spliceosome. Again, not on the study design, but if you want to go that far, go for it. And what it does after, so this process here that we've undergone is transcription, which is the last video that we covered. And then after transcription, we splice out the introns and join together the exons to create what we call mature mRNA. So this is mature mRNA. This is what we call pre-mRNA. And you can see we've added a 5' cap, we've added a poly A tail, and we have removed the introns. And this process here that we've undergone, this is stage 2 in protein synthesis. This is called RNA processing. I'm just going to, here's one other one to show you. It doesn't matter. Introns don't always have to be the same size. You can see in this one, the intron is actually really quite large. One of the introns is large that's removed. Now, the last thing I just want to really briefly cover is this thing called alternative splicing. So it's not on the study design, but always accepted by examiners. Yeah, if you write about alternative splicing, that's great. So we're now on page 7 in your notes. And what we've got here is that the splicing of mRNA in a different way. So sometimes you might cut out some of the exons as well. So you can see this mature mRNA has exons 1, 2, 4, 5, and 6, and it also cut out exon 3. Yeah, whereas this second piece of mRNA has exons 1, 3, 5, and 6. And what happens when you make these different pieces of DNA using this alternative splicing, which just means different combinations of exons, different combos of exons, is you end up with a whole host of different proteins that can be made. And we're going to talk about that when we talk about translation. So here's just one other image to show you what can happen during the process of alternative splicing. So what I'd like you to do is I'd like you to actually stop and complete the 2 exam practice questions. So it's exam practice question 6 on page 7 and 8 in your notes. Then in addition to doing that, you might want to do your 2B questions. Straight into Cambridge is fine, or you can do them using your hard copy of the book. And I'm just going to right now go and show you the questions that are in the book. So these are the 2 questions that I want you to complete. And then I'd like you to pause the video here because I'm about to go through the answers. So I'll let you pause. Now, hopefully you've restarted the video after you've attempted the questions. So I'm going to go really quickly through these answers. So the first one asked you to outline the events that happened during action X. So here is action X. Action X is showing me that we're talking about the process of transcription here. And we're going to outline those events. So straight from our notes, RNA polymerase is going to bind to the promoter region. It's then going to read or move along the template strand and join together free RNA nucleotides to create pre-mRNA. You could have also mentioned that the DNA was unzipping, but we're only looking at 2 marks. So I like these 3 points to kind of cover everything within that role. It then says, notice that, so question part 2 talks about P. Now, what is molecule P? If it's just after transcription, this is what I like to do. I like to annotate my diagram. So I would say that structure P is pre-mRNA. All right, so structure P is pre-mRNA. And so it has 2 different types of components. What are these components called and what happens to each of them? So this is an example where an RNA processing question is worth 2 marks. If you still haven't answered it, pause now, give it a go and see how you go. And this is what I would have written in this answer. I would have said that the 2 different parts are introns. These are the ones that are removed from the sequence, and the exons, these are the ones that are spliced together and they form mature mRNA. And that is all I want to say about RNA processing and just a little bit of a summary on transcription.